Hello everyone, today we are going to see the DNA sequencing technology. DNA sequencing means determination of nucleotide or base sequence of DNA. We have several different methods for the DNA sequencing: Maxham-Gilbert, Sanger sequencing, automated DNA sequencing, pyro sequencing, Roche 454, solid sequencing, and Illumina sequencing. So today we are going to see about Sanger sequencing, that is also known as dideoxy chain termination method. In case of Sanger sequencing, it involves the basic principle, that is use of dideoxy nucleotide triphosphate. This dideoxy nucleotide triphosphate, they differ from the normal nucleotide triphosphate, that is they lack the 3' OH group. As we know, for the normal DNA synthesis, the DNA polymerase always require the 3' OH group as a starting point for the addition of new nucleotide. And because this deoxynucleotide, they are lacking this group, the further addition of deoxynucleotide is not possible whenever dideoxynucleotide get added within the DNA chain. So based on this, the Sanger sequencing, it determines the nucleotide sequence of DNA during DNA synthesis. So for its sequencing, the basic component required, that is Sanger sequencing, that is first, that is single stranded DNA template as a template whose sequence we have to determine. DNA primer, which provides the 3' OH group for the first 3' OH group for the addition of new nucleotide by DNA polymerase. Third one, the DNA polymerase enzyme for the addition of nucleotide. 4. Radio label deoxynucleotides as a substrate for the DNA synthesis. And 4. Dideoxynucleotide as a chain terminator, which we have to add in separate reaction tubes. In case of Sanger sequencing, we are preparing 4 separate sequencing reactions. In each of these 4 separate sequencing reactions, we are adding same DNA template. Okay. After that, we are adding the 4 standard radio labeled normal deoxynucleotide in each tube for the addition of nucleotide by the DNA polymerase. Then we are adding the DNA polymerase as an enzyme to carry out DNA synthesis, and the DNA primer to provide 3' OH group for the DNA polymerase as a starting point for addition of new nucleotide. Out of this reaction, in each reaction we are adding only one type of dideoxynucleotide. So all the 4 dideoxynucleotide we are not using at the same time. In each reaction we are adding only one of the dideoxy. So like this, we are keeping 4 separate reactions. In first tube we are adding dideoxyadenosine triphosphate. In second tube we are adding dideoxyguanosine triphosphate. In third tube we are adding dideoxycytidine triphosphate, and in fourth tube we are adding dideoxythymidine triphosphate. So like this, different dideoxynucleotides we are adding in different 4 separate reactions. Once we have added 4 dideoxynucleotides, during the DNA synthesis, whenever dideoxynucleotide triphosphate get added into the DNA chain, it causes the termination of DNA synthesis, and we will get, because of that, the DNA fragment of varying length, which are terminated at point whenever dideoxynucleotide is get added within the DNA chain. After the synthesis and the addition of dideoxynucleotide, we are loading the product from the 4 separate reactions into the adjacent wells of same polyacrylamide. And after we got the separation of the different DNA fragment according to their band size on the gel, the relative position of different bands among the 4 lengths are compared, and the DNA sequence is determined based on the reading of the band from bottom to the top of the gel. Here the gel is shown, which contain 4 different wells, and in each of these 4 wells we have added reaction product from different reaction tubes. In the first wave, the A indicate here the addition of dideoxyadenosine triphosphate in the during DNA synthesis. C indicate addition of dideoxycytidine triphosphate. G indicate addition of dideoxyguanosine triphosphate. So in the adjacent wells of the same gel, we are adding the reaction product for these reactions. Once we got the separation of the DNA bands according to their length, we will compare the DNA bands in adjacent well. First we will determine which one is the lowest band and in which well it is found. So in that case, our lowest band is found in a well, that is G well. In that well, all the DNA products, they contain the DNA length that is got terminated whenever dideoxyguanosine triphosphate has been added. So first sequence we have got, that is G nucleotide. The second highest band we got in the N way. So here the next nucleotide according to the addition of dideoxyadenosine triphosphate is there. So next nucleotide is A is there. In the third band we got in the C well. So the next nucleotide, our next nucleotide is C, due to where the length, this length of band is produced due to the addition of dideoxycytidine triphosphate. So like this we are determining the sequence of DNA based on the addition of dideoxynucleotide, and based on this sequence we are determining the sequence of our template. The template sequence is exactly complementary to the sequence of dideoxynucleotide terminated chains. So based on that we are determining the sequence of our input template DNA in the Sanger sequencing. So Sanger sequencing mainly involves the use of dideoxynucleotide, which causes DNA synthesis to terminate due to addition of this dideoxynucleotide, which are lacking 3' molecules. I hope you have understood very well about the Sanger sequencing and the principle behind the Sanger sequencing, that is use of 3' OH group.